I'm Gene Robinson, and I'm privileged to be able to co-host this, co-moderate this next session with Kirsten Lindman-Toh. This is going to be a very interesting session. We have many, many talks, and we were faced with how we're going to deal with something like this. We could have restricted the number of talks. Instead, we decided to appeal to your better instincts as speakers. We're going to have a large number of very short talks, and these are all going to be progress reports on the various communities and the activities that are going on. Think of this as kind of a mellow data blitz, but think of it as a data blitz. So there will be an end to the talks, and they are fixed at the size of the talk, the length that we have indicated. So for speakers that are speaking for 15 minutes, you'll have 12 minutes. You'll hear an alarm, and then 3 minutes to wrap up or to take questions. And then the final alarm will go on and will keep going on while you're moving off the podium, and we introduce the next speaker. For those speaking for 10 minutes, it'll be 8 plus 2. And don't feel bad if you don't finish your talk. These are progress reports, and we understand your progress is ongoing, and so think of it in that way. So our first talk on microbial eukaryotes is going to be given by Neil Hall from the Earlham Institute. Neil? Oh, I'm sorry. I wanted to make one more announcement. It's not coming out of your time. You have on your tables an important note. Please note the Earth BioGenome Project launch and workshop are embargoed until the 1st of November, 12:30 tomorrow. So please refrain from sharing any details of the meeting on social media, etc., until after this time. You've been warned.
Thank you, and thank you very much for inviting me to talk. I'll put my timer on.
Kirsten's got you.
Okay. So I'm not really going to give an update on a community project. I think my job here is just to raise awareness of microbial eukaryotes and potentially discuss what may be a strategy for addressing this in the Earth BioGenome Project. Some of the take-home message, I'll put them in early in case I get cut off, is really that most genetic diversity and certainly in gene complement is microbial, but not just bacteria, but in microbial eukaryotes. We have a huge problem that most of these eukaryotes, these microbes, are uncultivated and probably uncultivatable in any high throughput way. However, they are hugely important, and if you take anything away from this, they are the basis of many ecosystems. If we want, and one of the aims of this project is to get complete gene inventories, we have to address the microbial eukaryotes. And if we really want to understand the origin of life, and which I think we all do really, we need to understand these organisms. And as was pointed out right at the beginning, the number of species is basically unknown. You can look at even modern reviews of how many there are, and they're orders of magnitude different, from hundreds of thousands to hundreds of millions, but it's probably many millions. And I can give reasons for that. There are efforts ongoing, and there's a talk about the 10KB project just coming up, so I'm not going into great detail about it, especially because of the intimidating introduction. This project is looking at plants and protists, I think aiming for around 4,000 protists, and this is a really important project because it's aiming to sample the phylogenetic tree, so we have at least in each grouping a representative species, but it's based on cultured samples for good reason, because that's how you can get long reads. But that means you're going to bias your sampling to heterotrophs, and also you're only ever going to scratch the surface of all the diversity that's out there. Now, these are, and I'm going to try and see if I can get this video to work, so that if you get bored, just watch the video, and you can see how beautiful these organisms are. So as we're doing lots of physics references, these are the dark matter of biology. So for the rest of the day, you're going to hear mainly about this branch of the phylogenetic tree, where the animals live, or possibly the fungi or the plants. But all of these branches, this phylogenetic tree, are equally as diverse as those branches of those phyla. And these phyla, this is probably incomplete. This is Sandy Baldov's tree of life. But all of these branches are wonderfully interesting. The Dictystelia and the amoebas that live here, they're hugely important to soil ecosystems. You have the alveolar, which are important to marine ecosystems, but also you have the apicomplexa, which are the important parasites that give rise to diseases like malaria. But the red dots I've shown is where we have some genomic information in the databases, but there's many where you have, such as the Rosaria, for example, where there's almost none whatsoever. And just to point out, so this is the representation of genomes in gold. This is somewhat out of date, but it hasn't changed that much in proportion. Whereas if you look at this representation of 18S sequences, you can see the vast majority of life is probably protozoan. And they're beautiful. That's the other thing that you've got to take home from that. So I can't change my slide. The other thing is, and this is a tree, some figure from the same paper which shows the branches of life, and this is a similar tree of life. And the outer ring, just where the dots are, is where there is some kind of sample that is in culture. And you can see for large parts of the tree, there's entire phyla where there isn't a culture available. And if you imagine that we didn't have a sample species from an animal or a fungi or a plant, that's kind of where our knowledge is. It's inconceivable. So how do we capture this diversity? And just to say, the way to go is to move from culture-free approaches, and instead of looking and trying to sample the tree, because that's going to be impossible, you need to move to environments, whether it be fresh water environments such as this, or whether you look in the guts of cockroaches, which actually have an amazing ecosystem of protists living in there. And I hadn't heard of the term, the Google Life, but I will change my slides accordingly in the future to refer to that. So I've been working with collaborator Tom Richards on strategies for how we can mine and sample protists in a high throughput manner. And he's been got funding fortuitously from the Moore Foundation. And this is a cell sorter, which is being loaded onto a ship, which is going out into the Pacific Ocean, so they can look at osmotrophic environments. They can take fresh water, put it through a cell sorter, and separate populations. So they're using markers to separate things with flagella from things which have chitin cell walls, things which might have chloroplasts, so you can separate the populations out and get single cells, which you can then amplify and sequence. And then you can look at how your sequencing that you're getting back is representing the diversity of known eukaryotic life. And you can see in this, they're missing entire orders of streptomenophiles, but getting good representation of different lineages based on which markers that they're using. And this means that you can get some idea also of what's coming through the cell sorter and then getting genomic data back. Now, you can tailor this approach to different environments. You can look at other cellular characteristics such as size or shape on some of the cell sorters you have now. And you can use different surface markers to sample them. And now there are approaches where you can augment the straight shotgun approaches with things like Hi-C and metagenomic analysis. And instead of what we've got from studies like this, which is like a bag of bits, not like the VGB standard, I know, but we've got to try and augment this approach with other approaches to improve it. So one of the things is this generating reference genome. So these are the data that I got out of NCBI not long ago around, if we go to orders, there's around 46 done to date out of 330. If you go to families, 79 of nearly 2,000. But if we can deeply sample environments, now, so this is a study where they looked at 18S sequences of all sampled freshwater environments at 18S level. And if you could get that environment, you could generate potentially 100,000 genomes. You'd get to your 1.5 million really quickly and be able to say we're done and forget the bats and the birds. So if we can solve this problem, it could be really interesting and really create a huge amount of information from where there's basically none at the moment. So it's really dependent on us being able to adapt these low input approaches So if you're going from a single cell, it's really, really difficult to get long reads, but the people are making progress in that area. There's a lovely study that's been done to demonstrating the power of using high C in populations where you have mixed microbial and eukaryotic organisms. So not only does it tell you about proximity within a chromosome, you can sort of say where a piece of DNA came. So if you can do high C, then you can start to get your clouds of reads and pull them into different single cells. And then the microbial, the bacterial people have made huge strides in metagenomic assembly using things like k-mer composition, taxonomic classification. But I think the secret is really going to be very, very, very deep sequencing. A lot of the studies that have been done on SAGs to date, they've been looking at a few plates of cells. If you went into an environment and sequenced hundreds of thousands or potentially millions of cells, then you start to see repeated sequences of the same thing. And from the work that I've looked at with Tom and that we've done, if you could do hundreds of thousands of cells, you could start pooling these data and generating not VGP standard, but respectable genome sequences. So why is this important? Like I say, most of biology is based around plants, metazoa, fungi, but we just haven't been looking. And if we could understand what genes are where in environments, in what cells, then we can start to do some very neat guilt by association studies about what genes occur in cells that have what processes and what genes co-occur, what genes are associated with what environments, and so how do they help environmental adaptation. Like I said before, things like malaria, sleeping sickness, and toxoplasma, etc., have been evolved out of these branches, many of which are really understudied, so the evolution of pathogenicity will come out of this data. Endosymbiosis has occurred multiple times throughout the phylogenetic tree, and understanding how that and HGT have given rise to modern eukaryotic life will be hugely, really exciting, and so that we can come back to the origin of life. So with the time I've got left, I want you to just admire these beautiful diatoms, which is one of my favorite protists. This is just one part of one of those branches. You see all of this diversity, so we shouldn't ignore it. Thank you.
Neil's starting us off really well. There's even time for a question.
Oh, really? Yeah, go.
Yeah, I was wondering.
Yeah.
In the beginning, you said you're doing cultured cells in order to get good long reads.
Yeah.
In our experience with the vertebrate genomes, that's not necessary. Although you will get longer fragments for, let's say, scaffolding, but even then, you can still get 200 to 300 kb fragments out of the tissue.
Yeah, but if you're going from one cell, it's really difficult to get along to actually extract DNA and get fragments, which would give you anything. Or you wouldn't be able to make a PacBio library from one cell.
No.
And I guess, and if you're amplifying from single cells, then you have all the problems that are with the biases that are. I mean, so as soon as you can get to like 100 cells, then amplification is doable because it evens out the biases. But from one, that you have a problem. But I think there are methods where you can mix sort of population sequencing with single cell approaches to generate, and we're trying it now, but which is showing promising results.
Yeah. Do you have any specific environments that you think are grossly undersampled and that will have hidden gems?
Well, unfortunately, so people, certainly when they're doing SAGs, have looked at freshwater or oligotrophic environments because it's really easy to put through a cell sorter. So we're still looking under the lamppost. But I mean, for example, soil environments are going to be hugely diverse, but they're understudied, and that is because they are difficult to get individual cells from. So, I mean, and like I say, things like, depending on what you're interested in, some of the arthropod guts have some really interesting metabolic processes, which are still despite the fact that people have been tried to study them using bulk approaches, are still not very well understood. So